Okay, great. Can you all see my screen?
Yes.
Yes.
Excellent. All right, so welcome. So I'm going to be talking about a topic that has been kind of like bubbling under the surface for a number of years, probably even longer now. And today we're going to try and address this overarching question in which we have a large number, an increasing number of entities that have essentially dual representations. These entities are things like human genes that are represented in existing databases and knowledge bases. Many of these have been around for decades. This includes big genomics resources such as Ensemble, NCBI, which is itself a large collection of different databases, knowledge bases, modular databases, and so on. And in addition to that, we're starting to see some of these entities being represented directly within obo-ontologies. And this situation has really kind of evolved over time rather than being something that was there in the plan for obo at the beginning. And it's evolved for good reasons. But, I think the time has come where we actually need a bit more of a top-down plan for how we organize all of this. How do we kind of like collectively, understand the requirements for the users for some of these things? How do we adapt our technical infrastructure to deal with this? There's actually a number of quite difficult knowledge representation challenges when it comes to many of these entities, and it also brings in particular questions above and beyond what we have for other ontologies when it comes to ownership and governance of these kinds of entities. So I just wanted to give a few acknowledgements kind of like upfront right at the beginning. So even though I'm speaking, really only for myself today, I'm building on the back of, the work of a huge number of people. I touched on a number of different projects in this talk, but this is primarily an obo talk, and I want to kind of like thank all the members of the obo operations committee, and a big special thank you to all members of the obo community, which I'm so happy to see many of you here today, and to an NIH grant to fund some of the infrastructure for obo, where some of these discussions initially happened. And so, yeah, and especially big thanks to Nico as well for helping get this seminar organized. So here's what we're going to do. So prior to this, many of you took part in a survey I put together, and thank you for doing that. Thank you for the patience as well. I mean, I think sometimes it's hard to kind of like pose some of these kind of like questions because they involve a lot of kind of like nuanced issues, but I think you all did a great job there. Thank you. So and there's still time if you haven't filled this in and you want to do this perhaps while you listen. Then this talk itself, will take hopefully around about 30 minutes or so, and we're going to be going over the historic perspective here. Just I think it really helps to look at the kind of history of obo and how we got here, and then we're going to talk about the kind of like the requirements, and then a framework for thinking about the problem, and then some challenges and next steps, and then we'll have a bit of time for discussion. But first of all, I want to just start with a bit of a historic perspective. So I think all of us on this call maybe remember the 1990s. So this is maybe a bit of a British-centric view of the 1990s here, with kind of like Oasis, Blur, and so on. But of particular relevance to me, that's Dolly the sheep in the bottom right-hand corner there. So this was the first animal to be cloned. Actually, when Dolly the sheep happened, I was actually sitting in the office next door to Ian Wilmot, who was the person in charge of that project. I had nothing to do with it. I was actually working on an animal genetics database in Edinburgh, Scotland at that time. And so I actually want to talk really about biological databases in the 90s as well. So when these were created, many of these were essentially silos. They were talking maybe about the same kinds of things, the same shared gene functions, shared phenotypes across these different databases, but they were, yeah, in large part siloed. And then, you know, starting with the gene ontology, ontologies emerged as a way of kind of annotating the entities in these databases. I don't have time for the full history. But I just want to give a kind of like a shout out to, you know, all of the, you know, the many different kind of like resources that kind of like were doing some of this even before the gene ontology, you know, the various different kind of biopsych databases and so on. So this is a somewhat simplified history, but I don't want to kind of like erase anyone's kind of like contributions before then. But, you know, I wanted to kind of like point at this obo paper from, you know, the first obo paper from 2003 that kind of like really laid out the kind of like the initial groundwork for obo, which was, you know, to come up with a set of descriptive descriptors for annotating objects in different databases that would allow you to do cross-database querying and use some of the same terms, same descriptive terms to essentially annotate and describe some of the entities in these databases. And so some specific examples, you know, of course, you know, I'm part of the Gene Ontology project, have been for a very long time. So Go is, you know, one of the canonical examples here. And, you know, Go from the outset was cross-species. It was used by both kind of gene and protein databases. And the basic idea here is, you know, for various database entities such as, you know, the patch1 gene in mouse, represented over here on the left in the MGI database with an MGI identifier. This could be tagged essentially with a term or descriptor from the Go, transmembrane signaling receptor activity, which has this Go ID, and different people could use that same Go ID even if they were describing entities in different databases or indeed in different species. We also have, you know, another early ontology was the sequence ontology for describing the types of different sequence features. So these are the different elements that can be located along a genome given coordinates. There was a need for a vocabulary to describe precisely what these kinds of entities were, you know, be they exons, introns, transcripts, stop codons, regulatory regions, repeats, all of the rest of it. So the individual entities remained with the province of the databases themselves, the individual exons, introns, and so on. But the descriptors were in the sequence ontology. The sequence ontology, you know, just as the gene ontology doesn't include actual genes, the sequence ontology doesn't include actual sequences. You know, their core early use case was phenotype annotation with some of the original phenotype ontologies, such as MP, mammalian phenotype, you know, other species such as TO for plants. You know, later on, you know, other ontologies such as the human phenotype ontology evolved. And then these were used to annotate a wide variety of different database objects, genes, proteins, variants, alleles, you know, entire organisms such as patients, also to annotate diseases as well. You know, expression annotation, again, descriptors that could be applied to gene, you know, database entities such as genes, proteins, transcripts, yeah, or indeed, you know, data such as samples and so on. And then also, you know, other ontologies for describing, not just directly describing these entities themselves, but for including evidence and provenance of all of the different kinds of associations. And this is still very much a key use case for obo. You know, if you look at, you know, any of the pages for, you know, many of these kind of like resources or different kinds of, you know, integrative resources or portals, you know, you'll see, you know, around the kind of like the core entities such as the genes and proteins, you know, you'll see kind of common themes such as, you know, essentially a list of, you know, different kinds of ontology annotations and other kinds of information about those entities, such as, you know, their phenotypes, their variants, their functions, and so on. And so, you know, this is still very much, you know, a key use case for obo. I think all of you on this call know exactly what obo is, but yeah, I'll give a brief summary. The basic idea was to have a set of well integrated and modular ontologies that were not developed as monoliths, but more as kind of like jigsaw pieces that could be assembled together to build this larger kind of like map of the set of all kind of like all the different ways of describing kind of like different kinds of biological entities. It was never designed to be purely technical. It's both technical and socio-technological framework for allowing people to come together and solve some of these problems. So we provide not just tools but also best practices and as well as infrastructure for forging new ontologies. And this, the basic idea is we can piece all this together and we can use it to curate all of the different aspects of all of the different things. So I'm not going to go too much into detail on some new developments in obo. I really like this kind of like forum for those of us who are more directly involved in obo to kind of like reach out to the broader community and tell you what's happening. I think many of you are aware already of this newer project called the obo dashboard, which allows us to actually more quantitatively evaluate different ontologies against the set of different obo principles. So I don't have time to go into this in this talk, but big shout out to Nico, Becky, James, all the people involved in developing this, and I'll be referencing this kind of like briefly during the talk. But I think it might make sense to have even another one of these community calls where we go into some more detail here. Okay, so far, this, what I've described, there's definitely kind of like quirks and things to be worked out, but overall, I think we're doing, we made a lot of good progress here, thanks a large part to you all in the community. But the kind of story I told you there was maybe a little bit of a simplification. And in fact, while we kind of like have made a big deal about orthogonality within obo, such that we try and make things such that the ontologies are orthogonal to one another, it's really worth pointing out that many of these ontologies actually lack orthogonality to existing databases. The same entities are represented in both external databases and in obo. And sometimes the same IDs are used, but in most cases, different IDs are minted. And so, maybe this is well known in certain domains, such as, chemical entities that will, Chembl PubChem will have entries for specific chemical entities, things that you can attach an INCHI key to. Kebi will have those entities as well as like very useful classifications of those entities. But we see there's, a number of other kinds of entities that have these dual representations, even for something like the Go, we see there's separate databases of reactions and pathways and so on. And then this area in gray in the middle is in some sense kind of like unclaimed territory, but I think we're going to start to see more and more ontologies essentially emerging to fill this gap, because we didn't arrive at this situation just randomly. There was actually a kind of like a genuine need for creating these kinds of ontology representations of these entities. And I'll go into a couple of these. So the ones highlighted in bold. So one reason is that there's a real use case for being able to reference things such as genes and proteins and chemical entities from existing ontologies. There's other kind of like drivers as well, such as like having essentially unified semantic representations of knowledge. And I won't really touch on some of the other use cases, such as like the need for named entities for text mining. Big resources like UMLS have had many of these kinds of things in them for a while. But there may also be an element of kind of, we're just doing this because we can. And so I think it's worth kind of like, looking at this and actually being really driven by requirements here and actually looking at some of the trade-offs. And, some of this actually may be hard to do at scale, involves a lot of duplicative effort in some cases with existing databases. it does introduce some confusion with our users, and there's a certain kind of trying to fit a round peg into a square hole aspect of this, and that maybe ontologies aren't, we can, many of us have a tendency to see ontologies as a hammer and, everything is a nail, but, they may not always be the most appropriate solution. But just to kind of like go over some of the requirements here and why some of these kind of like ontologies kind of existed in the first place. So, we can see, one of the original kind of like shortly after Pro was first developed, we saw, there was a real kind of key use case here, driven by this, I think this quite seminal paper led by Anne-Marie Massy. So this was, the cell ontology had existed for a while, but to define kind of immune cell types that were coming from like FACS experiments, we needed to be able to define cell types in terms of their surface markers. We wanted to define them not just in terms of text definitions, but actually using kind of like logical axioms so that we could use automated OWL classification and so on. So this diagram from the paper shows the kind of schema from the original dendritic cell ontology, representing classifying cells in terms of their proteins on the plasma membrane using protein ontology. And this need continues to exist. And what we're actually starting to see with now, with all of the the explosion of data in single cell transcriptomics is the need for the need to do this not just with proteins, but also with genes or transcripts as well, as we're discovering new cell types that that can only be defined by in terms of like the genes or transcripts that they express. So there's a nice paper here from this group on some of these requirements. And then in some ways these requirements can be transitive. We're starting to see within the protein ontology, these proteins need to be linked to the underlying genes. So what you can see here is in Pro that there's actually a number of classes in Pro now representing genes in different species. There's a class in Pro for sonic hedgehog in human, mouse, and chicken, zebrafish. And because many of these, they don't have existing ontology representations in URIs, so Pro have started using kind of like the URIs for essentially for the web portals for these resources for inclusion. So there's and these are based, of course, off of either the authoritative model organism database ID or NCBI gene or HGNC, depending on the species. So chemicals were also previously represented in existing databases, but we needed, we've always needed ontological representations of chemicals. Mainly because we need the classification that that doesn't exist in these databases as well. And so CABI provides a very nice kind of classification of these kinds of entities. We're making heavy use of this within the Go. Another example is organism taxa. These have existed in databases such as the NCBI taxonomy database for a while, but we've needed a kind of like ontological representation, for a number of reasons, including the ability to place taxon constraints in terms such as Go and anatomy ontologies like Ubron. So I just want to talk briefly about the NCBI taxon ontology within OBO. Many of you may not be fully aware of its, how it's actually created. Some people come to us and they say, oh, can you add, can you add this taxon into NCBI taxon? And of course, we don't actually do that ourselves. All we do is provide a very kind of like simple direct transform of everything in the NCBI taxon database. This is kind of like essentially run by a group of us within OBO. All the content changes come from NCBI. We don't layer anything on top of that ourselves. So you see a complete one-to-one representation of everything there. Now this leads to things that are not very OBO-esque in some ways, like the root of the ontology is literally called root. which is quite an odd thing to do from an obo perspective. You know, we use, it's conventional to use plurals in this database, so we don't stem these plurals. We just say cellular organisms rather than cellular organism. We use capitalization. So all of these actually go against obo principles, but you know, we're just trying to keep it simple and have a direct transformation. And then there's definitely some kind of like, you know, things in here that violate the scope constraint of obo as well. For very practical reasons, NCBI includes metagenomes within the hierarchy too. And these just come along for the ride with our direct transformation as well. And, you know, maybe that shouldn't be the case, but we partly just as a result of the lack of resources, we just keep it simple and we translate everything that is in the database, even if this results in things like biofilter metagenome being placed in a taxonomy ontology. And then there's a number of other drivers for essentially turning databases into semantic resources as well. So this kind of seminal paper from Ruttenberg et al. Was kind of like a manifesto of how RDF-AL could be used as a framework for unifying different databases. And it's definitely, you know, very attractive to use this framework. It gives us a unified way of representing graphs, of having unambiguous URIs for each of the elements in this graph, as well as a precise semantics as well. Although having a translation doesn't necessarily necessitate using an ontology, but many of these entities, such as genes and so on, are very naturally represented as OWL classes. So to recap so far, you know, there's a number of different use cases for re-representing these entities from existing databases in ontologies. The need to axiomatize, the need to essentially have a unified semantic representation of knowledge. There's different ways of doing this. There's things like the NCBITaxon approach, where we do an automated translation. There's approaches such as Pro, which does a more kind of like manual ontologization approach. There's still potentially a lot of confusion with what we're doing here with these different representations of these databases. So I thought next we could go over a kind of like a framework for looking at these kinds of distinctions. So when I draw this distinction between databases and ontologies, it's not really intended to be some kind of like crisp binary distinction. It's more of a spectrum, or really a kind of like a collection of different features where different things can have ontology-like aspects and database-like aspects. One of these criteria is essentially the scale. So ontologies have traditionally been on the smaller side, with something on the order of kind of like thousands or tens of thousands of concepts. Something like Go is often considered a larger ontology with like some 45,000 terms, whereas databases tend to be a little bit less bounded in terms of like the number of entities. And we can see this with these stats from Obo that show the kind of like the top and the bottom kind of ontologies in terms of like the number of entities that they have. And indeed we see, databases that are recapitulating, ontologies that are recapitulating databases amongst the kind of like, having the most classes in Obo. So NCBITaxon, Drawn, Drug Ontology, Pro, KEBI, and so on. But even these are kind of like, dwarfed by the actual number of entities that we see in some of these databases. So KEBI, even though it's one of the larger Obo ontologies, only includes a fraction of the actual entities that are in something like Campbell. The protein ontology is like a very large Obo ontology, but it's more like approximating the number of entries in SwissProt rather than say all of Tremble. And in fact, it's still, the coverage is still quite a bit below SwissProt, because many of these classes may represent different kind of like phosphorylated forms and so on. And then even though we have ontologies of kind of like genes, OG, within Obo, there are databases out there, such as this is the IMG metagenomics database at my own institute. This currently has 65 billion genes within it. And that would be a little bit of a stretch to try and include these in one ontology. So another kind of defining characteristic is the nature of the hierarchy. with ontologies, typically have classically been regarded as being kind of like hierarchical in nature and having a kind of like a rich is a classification. So maybe taking HPO as kind of a canonical ontology, you see that the class within HPO that has the most direct children, abnormal circulating protein concentration, has 56 children. On average, things have like more like, I know, 5, 6, 7 children. But then if we look at some of these more database-like ontologies, you'll see classes that have something like kind of like 20,000 direct children. Indeed, in drawn, there's one class that has, it's very flat. There's one class that has like 500,000 direct children. And so there's nothing, inherently wrong with this. I think, you shouldn't have a hierarchy where one doesn't exist naturally. But I think it does put a strain on the kind of like the conception of what an ontology should be used for. And this can be seen actually in some of the tools. If you try and open some of these things within OLS, sometimes you get this kind of like spinning wheel of death as it churns around, before crashing your browser, trying to put like 20,000 direct children with under that. That's not the fault of the OLS or necessarily the fault of this, the ontology, in this case, OG, but it's more just a mismatch of expectations here. So I'll talk about this one in more detail later on. Another kind of like maybe controversial feature here is that I think some of these databases are more clearly delineated entities, whereas ontologies are more for our kind of like our own kind of like human construct way of trying to kind of like figure out how to classify these different things. Ontologies typically, especially within OBO, should really have textual definitions, whereas this text definitions don't really apply, I think, so much to things that are in databases. And sometimes, what we tend to see is, users prefer, more descriptive text. So this is like, some descriptive text about the human hedgehog protein or gene, whereas in Pro, this is represented more as a classic kind of ontology definition. And this actually kind of like shows up when we look at the dashboard report for things like NCBITaxon, which has no definitions, and also for KEBI, which has a pretty large number of missing definitions as well as duplicate definitions. I don't think these are necessarily the fault of the ontologies or the underlying databases themselves. It's more a fact of, we're trying to kind of fit a square peg into a round hole here. And we're trying to apply ontology criteria onto things that are more naturally databases. So I'll just kind of quickly go through some of the others. We strive for orthogonality within OBO, but there are just naturally, in the database realm, often kind of like duplicative databases for any one kind of entity. But of course, we have many things in common. Both are amenable to a semantic graph-based representation. They are kind of like curated from the literature. But I think there's definitely a kind of like a square peg round hole thing going on here when we try and take these databases and we try and make kind of ontologies from them and we try and apply OBO criteria to them. So it really kind of like poses the question, are some of the structures and the principles that we've developed for OBO, do they really apply here? So I want to go in a bit more detail just about this kind of like what I call the knowledge representation quandary. So ontologies, themselves arose out of a field that prior to ontologies was known as knowledge representation. OWL description logic is the main representation language these days for ontologies, but yet class-based OWL ontologies actually have quite definite limits on knowledge representation. So ontologies work, spectacularly well for certain kinds of invariant knowledge, and I chose the Go here to exemplify this. So the Go is encoded using OWL. So we actually have these, what many people think of edges are actually OWL axioms that express invariant knowledge. So we say things like, nucleus is a subclass of intracellular membrane bound organelle. Intracellular membrane bound organelle is part of some cell. So now you could argue that this is not, these axioms don't actually represent things that are truly invariant. Nuclei could be extruded from cells, But because we essentially own the conceptualization here, we can say, well, that's not what we mean by a GO-nucleus. What we're talking about here are canonical nuclei that are found within cells, and these rules that we specify essentially always apply. They are in one sense invariant. But there are many kinds of biological knowledge that don't have this kind of invariant property that you can distill into simple all-sum axioms that always hold to be true. This includes kind of like edges such as between a disease and a phenotypic feature, say holoprosencephaly and cyclopia, where it's not the case that you can write all instances of holoprosencephaly has feature some cyclopia, because there is variable penetrance for this disease. There's variable severity as well. It's actually quite hard to capture some of these things using OWL. You can kind of cheat and just make OWL axioms, which gives you a nice kind of structure you can traverse over, but it's not really clear that is the best use of OWL and the best way of encoding knowledge in an ontology. So you don't have to just take my word for this. I regard kind of like Alan Rector as one of the giants in the field, so as well as some of the others on this paper. And so this is a really nice paper from a couple of years ago. Alan actually gave a wonderful ICBO keynote on this exact topic. So, I don't necessarily agree with everything in this paper, but I think it's a really kind of like good summary of some of the challenges in knowledge representation and ontologies. And I'd recommend this for all of you to read. And I'm indeed taking kind of some of the language here, such as about invariance and so on, from this paper. So I'll discuss briefly some of the kind of like the alternatives that we've come up with in different projects. So I'll talk briefly here about GoCams, causal activity models. So this is essentially next generation annotation within the gene ontology. So we're going beyond just simple standard annotations where we tag genes with terms, according to the original model that I laid out earlier. We're trying to kind of tell a more detailed story about how genes work together in the context of pathways to carry out their different functions, which are causally connected. So we do actually under the hood use an OWL representation for GoCams, but we absolutely do not consider a GoCam as an ontology. And we get around this kind of like invariant knowledge representation problem by essentially having more of an instance-based representation. So under the hood, what we're saying here is that, there is some instance of the ZIP2 C. elegans gene product that has DNA binding transcription factor activity that regulates the activity of the IRG2 gene product, for example. We're actually making use of a number of different obo-ontologies here. The Go itself, other ontologies for providing kind of like cellular anatomical context, such as CL and Uberon. We actually we ended up making essentially a very simple kind of like flat ontology for essentially as what Alan Rector calls a kind of conceptual coat rack to hang this more detailed knowledge off. So we made an ontology called Neo that is not something we intend to submit to Obo. It's really just there to be able to kind of like hang some of our gene functions off of, and that's essentially because we needed a representation of genes and proteins across a very wide range of species going beyond what is in Pro, and people wanted to use the identifiers from their database of interest. So using kind of MGIDs, using ZFIN IDs, and so on. And we actually, as part of this, we actually take Reactome as well, and Reactome has their own kind of essentially a database of different entities, and we make a translation of this called Reacto as well. So another approach is this emerging area called knowledge graphs. Now the term knowledge graphs is a bit buzzwordy, a bit of a marketing term. In many ways, you could look at knowledge graph and say, wait, this is what we've been doing all along with ontologies. But I think there is actually one key difference, and that because knowledge graphs aren't using OWL, and they are from the outset not intended to be the same kinds of artifacts that that you would find within ontologies. In particular, edges within a knowledge graph can be a lot looser than edges within an ontology. So they are more suited for being able to kind of like associate things like one protein with another with an interaction relationship, where you can kind of essentially tag that edge with properties such as its affinity. You could also even have probabilistic associations, or associations, for example, between disease and phenotype, where you annotate these with frequency and severity. So you can represent all of this in an ontology if you want to, but it does pose a number of problems, such as the ones that are highlighted in the Rector et al. Paper. And so I've got a blog article about some of this as well in the link below. And so there are, in fact, a number of emerging different kind of like biological knowledge graph projects. These include things such as Wikidata, which is, many people are familiar with Wikipedia, but Wikidata is in fact a big, large community collaborative knowledge graph that is semi-automatically constructed using open information from existing databases. They essentially kind of mint their own IDs for things rather than kind of using existing ones. But it's a really interesting project that I think I'd urge you all to take a look at. There's a number of other different kind of like knowledge graph projects there. Daniel Himmelstein's HeteoNet, the UCSF Spoke project. I myself am involved in things like the Monarch knowledge graph, which is itself part of a larger, being used as part of a larger NCATS funded project called the Biomedical Data Translator, that is essentially trying to unify a number of different knowledge graphs. And this is, to a large extent, even though these make use of obo-ontologies, they also kind of like include automated translations from existing biological databases. And just by using this kind of like looser way of being able to associate things together, we can do this in such a way that we get a lot broader coverage than some of the existing obo-ontologies, like like Kebi, for example, which only contains a subset of of all of the chemical space. And there's a number of emerging projects that I don't have time to go into today. This kind of concept of a KG hub, where we're trying to essentially kind of coordinate all of the different efforts here, in a way that is kind of like in part inspired by the obo effort to coordinate ontologies. But the scope here is really, really much broader with a lot more entities. So, I think the question for obo is, what is our path forward for how we treat essentially the ontologization of some of these different databases or knowledge bases? I've kind of like scoped what I think are kind of like three possible paths. We actually may end up having some kind of like hybrid of these, or different kinds of entities may call for different kinds of solutions. But I think it kind of helps to kind of like paint kind of broad brushstrokes like this. So, one model may be the not-in model, where you say essentially obo is really just strictly scoped to kind of like classic ontologies that, on the kind of like one end of the spectrum I showed earlier. And then we say, okay, but obo, many of the things in obo are successful, but let's encourage the development of some kind of like parallel obo for some of these other efforts. Then the other extreme is kind of like what I call the all-in model. And here, in obo, we explicitly expand our scope and say, okay, we're just gonna bite the bullet here, and we're gonna say everything is in scope. Introns, exons, if someone wants these to be represented in an ontology, we'll take them in. But I think there's many questions that need to be answered where we do this in a coherent model, in a coherent way. And then there's more of a kind of like hybrid. And yes, and thank you for marking that, Matt. I think the key thing is we shouldn't just do this in a kind of like ad hoc way based on who decides to submit the ontology first, but we need to do this in an intentional way, with taking into account not just the broader obo community partly represented in this call, but some of these these other databases as well. Matt, do you want to remove your your red squiggle on the screen there? I don't know if I can do that. So, you know, some of the kind of like the questions I think we need to answer are, you know, which entity types are a priority for this kind of like, you know, either for a kind of semantic representation, but also for inclusion in OBO, if indeed we include these at all. You know, how should we construct these? Should we go with a methodology where we do a direct like just as we did with NCBITaxon, a direct transformation of a single resource, or do we merge together multiple resources, or do we just kind of expand kind of ontologist resources and essentially kind of like create some of these things de novo, or, you know, do we have some kind of like hybrid model for the identifiers and the URIs? Do we mint, you know, completely new ones? Do we reuse IDs that already exist and are well used within the broader, you know, genomics community from these, or do we have some kind of like mixture? The URIs themselves, you know, do we use obo pearls? You know, there's emerging systems like identifiers.org that attempts to provide standard URIs for all major resources, databases, and knowledge bases. You know, in some cases, some databases such as UniProt already provide semantic pearls. Should we be reusing some of these, or do we just use the URLs that are provided in the existing web portals? A really tricky one is ownership and governance. You know, we could just have a model whereby whoever submits it to obo wins. So this means like, you know, postdoc who writes a Python script to say, convert all of Ensemble into OWL and submit this to obo. Do we accept that? It's probably not the approach we want to be taking, but, you know, how exactly should we tackle this problem? Should this be, you know, something the obo technical group does? Should the resource themselves provide that? You know, many of them don't actually have the funds to do this kind of thing, and it's not, to be frank, it's not really something that is of great interest to the majority of their communities. And, you know, maybe we have some kind of like a hybrid model here. So what I propose next steps is, this is really just the beginning of a discussion. I think, you know, as well as, you know, many of you on this call, some of you wear hats where you represent some of these existing resources, but I think there needs to be a kind of like, you know, a broader conversation that's maybe kind of like pitched at a higher level, where we don't talk so much about, you know, OWL invariants and so on. You know, maybe I think, you know, it might be nice to do a kind of like a white paper in this area. You know, funding for something like this will always be a challenge, but, you know, open to other ideas about how we progress with this. So I thought now we could just kind of like, you know, have some general discussion. Sorry, that went a bit longer than I intended, but we could also do a quick look at the summary as the survey as well. But before we do that, maybe I can just take some quick general questions from you all.
Hey, this is Asia. Actually, I suggest two funding opportunities for this kind of activity. I think to organize all the biomedical knowledge graph is a very good direction to go. And NIH just had established this Bridge to AI program, and the data generation project funding, what is called ROA, just released. And I asked Oliver to forward to this group because I don't have access to the obo discussion. I don't know if Oliver forwarded to you. I think this is a very good chance to get funding from there. And another opportunity of NIH funding is NIH now has the trans NIH knowledge base funding opportunities. So I think we as a community, we should do that. Should write a proposal to this either Bridge to AI program or the NIH trans knowledge base funding opportunities.
Great. No, I think those are fantastic suggestions. Yeah, I mean, I might suggest we as a community to kind of like agree first on what it is that we're we're seeking funding for, and maybe I think some of these are maybe quite timely, but I think maybe the first discussion to have is exactly, well, what if we had the funds, what exactly is it that we would do here? Would we write a bunch of kind of Python that would kind of like turn all these databases into ontologies, or is this more of a kind of like a problem that is kind of like partly kind of like social? Yeah, I don't. But what do others think?
Yeah,
and maybe let's bracket the kind of funding discussion a little bit until the end.
Yeah, definitely. I like Chris' talk a lot. Yeah, that's something I have been thinking also for years. But definitely, I'm not the kind of high scope. I've been working on the OGG, right? So ontology of genes and genomes. Yeah, for many years. Sometimes I spend a lot of time on it actually, and then I had a lot of problems. So it's very big scope, but definitely you cannot just automatically convert all the genes into ontology directly. So yeah, in ontology level, you need to have some kind of design, like very careful design. And in many cases, like for example, particularly for virus or bacteria, parasite, right? So before you have a lot of genomes, available, and then later on they put a reference genome. They say, okay, there is one like E. coli, like something reference genome. But the ID often used is still for the reference genome. It's not for the home like E. coli. So it's like you automatically change it. It's like the ID is for the reference genome, or it's really for the whole E. coli, right? For human, it's probably easier because you only have one genome. So, and then how to classify them into hierarchy, what relation to genome. So it's a lot of things. It's not as easy as you would think. The NCBI taxonomy is relatively easy because it's just single hierarchy, is a hierarchy for other things. I would think, we need to, it's like, I think, you have, Chris, you gave 3 options. I would think we can use the middle one, the partial in, but of course, it's just my personal thinking now. It's like you have to make one mature and then do it, and then make another one mature case by case and see how it goes. Yeah.
Absolutely. Yeah. And sorry, I had to give kind of quite, such a short summary of Og in that, I think there was a lot of material to cover, but very, very good points. Yeah. And I think, maybe a kind of key question there is just what are the user requirements there? Do we even need a hierarchy for these genes? But I see, David, you've had your hand up. And then
I suppose so. The use cases, having a good summary of use cases, I think would help a lot. And I think I could see, the use cases for having enormous OWL files are probably, I can see that that's potentially problematic. So another way to maybe phrase this, I'm wondering, or frame this, is in terms of an API endpoint. So, as someone building ontologies, there's frequently I need to be able to refer to a gene or gene product or the products of a gene generally, and I have a problem with, I've got some data coming in from Ensemble, I've got some stuff coming from a model organism database, I've got some things that are coming from CL, which uses Pro, and I want to be able to line all these things up. And I want to be able to track versions sometimes as well. I've got single cell RNA-seq data coming in, and it's all against a particular version of the mouse genome from Ensemble. Wouldn't this be much better suited to an API? You could imagine something built over something like MyGene, which says, I want to refer to this gene. I'm going to give the API a certain amount of information, that maybe the Ensemble build version, an identifier, that's my starting point. And then it mints, I don't know what kind of URIs we would use, but it then mints a record, which I can then, it doesn't need any structure. We don't need a whole ontology, because at most, maybe it gets classified as a gene or a transcript. And then, what I want from that is a whole lot of synonyms for a start. I want the users of whatever I build to be able to go and search in all the different synonyms of that gene. And I want a way to be able to refer to collectively, You know, in some cases, maybe I want to refer specifically to proteins or genes, but I want to just be able to say, here's something that refers to a gene or all of its products, and it pulls those things together for me and pulls a bunch of identifiers together for me. Do you think that makes sense as a way of framing it, rather than let's build an ontology? You know, in some cases, we're going to want some kind of OWL entity to refer to because we're building OWL artifacts.
Yeah, if I understand what you're saying, there would still be, you know, you would use this, but it would still make an OWL file that you have to include in your imports, or maybe I'm thinking about this.
Yeah, but we wouldn't be building, you know,
yeah.
an entire, it would be on a completely ad hoc basis as needed.
Yeah.
I mean, because to me, the only reason to build the whole structure at once is that you need some kind of hierarchy there. But if the hierarchy is just some very vanilla classification of entities under SO, and that's it, then
or SO plus species. But yeah, it is.
Yeah, SO plus species, sure. But then it just automatically pulls in all the metadata in a standard way, and then I don't need to worry about which identifier to use. It just lines everything up, puts all my xrefs in there, whatever. I can then, other people can use it easily to map to the identifiers they want to use for their analysis. Does that make sense?
Yeah. So but I still see it as maybe a slight variation on the kind of the automated translation approach. You know, you could imagine doing the same thing for NCBITaxon as well. I think very few people need the entire ontology as one single artifact, although it is useful to browse on the ontology browsers. Yeah. So Pankaj, you also had your hand up.
Yeah, hi, morning everybody. Sorry for coming in late, but I like, David, you gave a very nice spiel about that there is a need for making some consistency across various other resources, whoever is providing a data on a single object or single object type kind of things. And then a lot of it is what do you call it, as hidden in the metadata aspects as well, and how to gather all the information, align all the information. And another bigger problem is version numbers. And people I know that in the plant community, for example, they have stopped using the same IDs for a different version of the gene or a different annotation of the genome. So I think it's coming out to be that there is going to be a lot more noise and a lot more maintenance of the ontologies if we go that route. So fortunately, we are having a discussion in the plant genetic resources treaty that's what do you call it, as implemented worldwide on the seed material or genetic material that is exchanged all around. And so they came up with a solution about using DOIs for the germplasm entities, that anybody who's using your germplasm has to cite that DOI, and different databases and data structures don't need to change anything. All they need to do is use the citation indexing APIs to start using those things. So on that similar lines, what we were thinking about is we're calling things as, there was already a discussion about a digital sequence identifier, but I think that's very narrow. So because a lot of genes and things like that are not yet sequenced. So we were thinking about a digital genetic object, which is behind the scene is actually a citation index or a DOI assigned for it. And then it's the Crossref and other sources, they manage it, but there's obviously some databases would have assigned a DOIs for that. So that way people can use it more using the citation indexing in order to come across the same thing. They don't have to repeat and name a naming convention and all sorts of nomenclature. That may go away as well. So I'll share the paper with you guys, but let me know if you have any thoughts on that.
Thank you. Yeah. So just conscious of the time, I see Charlie, you had your hand up, and then Bjorn after that.
Yeah. So I'm gonna say something very heretical. I don't like IRIs, and I think that the IRI thing is becoming a big problem. Like you mentioned, should we use a UniProt Perl? Should we use an obo one? Should we use the IRIs that Ontology Lookup Service creates, which some of them are derived from obo Perls, which so this is a big problem, right? Is mapping between all this stuff and figuring out what's going on. So yeah, so I'll come back to that one second. I have written a bunch of these scripts, like you mentioned, in Python to dump a ridiculous number of resources that are databases that have identifiers, some with synonyms, some with hierarchies, some with even relationships that are RO compliant, some with relationships that don't fit within RO. And I have all sorts of scripts converting these things into, obo and owl. And I posted this a little while ago. So I'll share that link again one more time in the obo Slack chat, and I'll put it in the chat here before we go, using this piece of software called PyObo. And so, yeah, you can start dumping these things. I did this with PubChem Compound one time, and I think the file was like, on the scale of tens of gigabytes. So it's not really the kind of OWL file you want to be making. So yeah, it definitely depends. And then coming back to what David said, a lot of the things people want to use ontologies for, like a lot of those operations can be sort of excised from the whole OWL and ontology and obo community itself. Like looking up things, classes, and their entity types and their names and synonyms and xrefs. Like this fits well within the OWL metadata standard, but you can also do it without being an ontology person. So those are all sorts of things I'm thinking about at the same time. And then back to the IRI thing is that a lot of the OWL resources require you to refer to another OWL resource if you want to use its IRI for an entity. And I think that's a big limitation because as we're going right now, it means everybody has to make like an HGNC OWL document, and you have to make a PubChem OWL document. And there's a lot of good reasons why you should or shouldn't do that for different resources. So I come back to why I don't like IRIs. It's kind of forcing us to consider everything should have like this web location. And then if you think about like identifiers.org, I'm not a huge fan of that these days because I think it's very slow to stay up to date with things. And there's a lot of problems in identifiers.org, a lot of metadata inconsistencies, like stuff that needs to be curated again. I actually, if anyone wants to talk about that, I have a lot of thoughts on that. And I've done a lot of this curation to sort of normalize it and make it work with all the resources that just aren't there. So these are all things that we have to keep thinking about going ahead. That wasn't really a question or like a comment on that.
No, those excellent, great comments. Bjorn had his hand up as well, but he may have had to leave.
I don't think I could add anything useful that should make people stay on. So I think this is good discussion. Okay,
so I apologize for going slightly over time. I think we've got a lot of useful discussion to have. I would like to continue this conversation. I will kind of like, set something up in the future so we can continue this. I also just want to make sure if anyone is, what I suggest we also do is set up a Slack channel for this as well. If anyone is not on the obo Slack already, you can follow up with me. And I think we can just kind of get a document going, a Google Doc going that kind of like summarize some of these and maybe have some kind of follow-up call. Any other kind of quick questions, quick comments on how we should kind of proceed? Because I'm conscious, many people have to leave now. I have to leave as well. And I'll preserve this the chat as well.
So Chris, I think what you said earlier is that I think writing a position paper or something like that is going to be super useful. Nobody minds having another paper, but also like for us to really focus what we are really saying. I think it'll also help me then to read back of what really the background is and where we're going. I think I have some disagreeing thoughts, but it's going to be help put things into writing.
Good, yeah. Okay, so you should feel free to contact me if you need added to the obo Slack or anything, but I'll send a follow-up email as well when we have something going. And if you want to kind of like volunteer to help with this as well, then I don't mean to kind of like own this process, but please kind of go ahead and contact me, and we can I suggest we take the next steps to Slack. Any other parting thoughts before we go? If not, thank you all for coming, and thank you all for the survey as well. I'll kind of like send out a summary of the survey results too. All right.
Thank you,
everyone.
Thank you, Chris.
Thank you. Bye.
Thanks, Chris. Bye. All right.